Good job on the understanding of mitochondrial gases for species and the aging process. And let's welcome you.
Thank you everyone for being here and thank you for the invitation. And it's a total pleasure for me to be here, and especially I think today is a truly special day for me because both my mentors are sitting in the audience, and my PhD mentor, Dr. Hong Zhang, and my postdoc mentor, Dr. Andrew Dillon. And thank you, Wesley University, for making this happen. So for this talk, our lab is really interested in understanding how mitochondrial stress is involved in the aging process. And as individuals around the world living longer, I think one of the biggest problems is the rise of chronic diseases. So conditions like heart diseases, neurodegenerative diseases, metabolic disorders are becoming more common as we age. By understanding the molecular mechanism of aging, and we believe to find ways that we modulate the rate of aging, such as interventions like dietary restrictions, supplements such as metformin, and also exercises, related in ways to delay aging and age-related diseases. So this is the idea that we cannot need to actually treat specific chronic diseases, but in a way that we delay aging to prevent them from happening decades before they actually appear. So our lab is really interested in understanding mitochondrial function. So why is mitochondrial function measured in the aging process? That is because mitochondria aren't just only the energy factories. It is also crucial for responding to stresses, such as when mitochondria faces with relations such as oxidative stresses and disruption of photosynthesis stresses, and also nutrient operations. Mitochondria could activate these protective or defense mechanisms to cope with the organism's stress condition. And some of this stress response pathways actually help the organisms to counter the stress response along the aging processes. So we have to be aware that some of the level of the stresses could activate this protective response that promote longevity. However, if the stress is too severe, it actually overwhelms the organism, leading to accelerated aging. So here is the notion that I think the ability of the mitochondria to respond to stress actually is the key to health and longevity. And this picture actually illustrates our current understanding of this systematic perspective on mitochondrial stress and aging. So changes at a cellular level could alter the function of the tissue and the organ, which in turn could communicate with each other and in a way that could modulate the aging of the entire organism. So our peers work focused on understanding how the mitochondrial function is communicated with the nucleus to modulate this stress response and affect aging processes. So for today's talk, I will only talk about the inter-tissue mitochondrial stress communications and the way that we could identify the signaling molecules that can modulate this aging process. So our work was built up on the pioneer work from Dr. Andrew's work, and in his lab, the report revealed that very stressing phenotype mitochondrial perturbations associated with the neurons which communicate the stress signal to peripheral tissues. Their response top is really essential for the organisms to cope with the stress during aging. If we can only activate the mitochondrial function in a couple of neurons, actually usually lead to longevity phenotype. And building upon these fundamental discoveries that we set up, conduct the suppressor screens and identify very interesting morphogen, the secret signal wheat, specifically IL-20 with ligand with salience. That is involved in this neuronal to intestinal mitochondrial stress communication. And we tried to make this transgenic worms by making worms expressing this protein ligand, specifically in neurons. We could see these worms actually quite long-lived, and also with altered metabolic in the peripheral tissue, such as in intestines. To continue on this question, to identify what are the signaling molecules involved in this crosstalk. If we could find more, maybe we could have this interventional target, a secret signal that can make pregnancy longer. So here is the strategy that where we have these worms producing weight at the 28 with light-specific neurons. Now we could see these worms rapidly activate mitochondrial stress response pathway in their intestine. So we conduct this DNA suppressor screen where we got many suppressors here. It shows you that the suppressors we obtained from this really robust screen results. And one of the suppressors we got from this screen is encoding a gene of SRD75 that is specifically suppressed this neuronal intestinal crosstalk. And we found that actually this gene encodes a GPCR protein receptor that only expressed in a pair of sensory neurons, chemosensory neurons in cells. And we continue to dissect the molecular mechanism from that all these GPCR signaling pathway that are conserved in mammals that they are not signaling pathway that is essential for the interstitial crosstalk. And we try to specifically activate this GR signal in just two sensory neurons in we could see a lot of metabolic status actually change in certain tissues, such as mitochondria is activated in the intestine, and their protein stresses is improved in their muscle cells, as well as their less lipid accumulate in their intestine. So you could think that this one is probably our monkey because they look really healthy. But unfortunately, these ones are actually not monkey. They're big shortly because we actually considerably activate this protein signal in this neuron. So even though it seems like the healthy lifespan is improved, their overall lifespan is not changed. So along the same line, so we want to identify more neuronal signaling to see if we could find a cure. And recently we published another paper that identified a similar morphogen to wheat, which is a very canonical morphogen signal, which is TDF beta signaling pathway. And this TDF beta is also functioning in these two pair of neurons. Different from this GPCR pathway, it sits in this ASI sensory neuron. So that cell is the TDF beta ligand that is produced by the ASI neuron and secreted from the ASI neuron. That can act on the receptor that one on the interneurons to coordinate this neuronal to intestinal stress response. And also, you can see that the lipid metabolism is altered in their intestine as well, and also the animal has improved the pathogen resistance ability. And also, it's quite interesting to notice that this neuronal signaling pathway is actually regulated by the dopamine, neurotransmitter dopamine, but also negatively regulated by GABA. So, for instance, if we block the GABA signaling, overall mitochondrial activated in the and without really disrupt our neuronal function. So, indicating us there's definitely a lot of neuronal signals actually involved in different ways in modulating overall metabolic of the aging process. So this is our current understanding of how neurons coordinate distress with peripheral tissue. So for instance, that we can have different ways to induce mitochondrial dysfunction within neurons. And there are multiple secret signals including multiple weight and TGF beta, and also these are needed in peptide. They all involved in this protocol. And also, to our surprise, we identified the neurons actually communicate with germline, the oocyte, mitochondrial oocyte, to make them produce more mitochondrial DNA, so the offspring can inherit more, and their offspring can be a bit longer and have a stronger resistance to stress. So all this actually only focusing on the secreted signals. What we really want to know is what happens to neurons. How neurons actually respond to the mitochondrial stress to mediate this secret signal. This is come to this illustration that is a question that we would like to further answer is how does mitochondrial stress lead to neurotransmission? This always not a new question because the people working in neurology field, they know that it's very important it's mandatory for neurotransmission for sure. But all this acute stress, right, we change the synaptic transmission immediately. But aging is a long process. How about mitochondrial function decline gradually with aging? How does that actually modulate the sustained neurotransmission with age? Right? People haven't answered this question. Yeah. We want to use our system to dissect this single molecule. So again, now we take advantage of the genetic screening algorithms and conduct the suppressor screen. And we're so lucky to get another suppressor, a gene engineer that is called TMDM2. You can see that loss of this loss of mutation, TMDM2 shows a strong suppression of mitochondrial intestine in worms that is neuronal gene overexpression, and also it does not affect the intestine microbiota itself. So what is TMDM2? And TMDM2 encodes the protein belong to a very revolutionary concept of transmembrane fast-mediated motif-containing protein called TMDM. So here illustrates the 6 members of TMDM family in humans, as you can see from 1 to 6. And interestingly, all these TMDM proteins actually localize on different membranes at different orbits within the cells. So for instance, TMDM5 is specifically localized within mitochondria. And TMDM6 is on the R. And the one that we identified from all these TMDM2, actually, the majority of them are localized on the plasma membrane. And the basic function of this plasma membrane protein actually is linked to calcium homeostasis. So there's not much information about that, so we decided to further explore the function of this gene. And the current graduates, the former graduates, now graduated already, but Joshua in the lab took this project and wanted to answer how does TMDM2 in the neurons actually mediate this neuronal mitochondrial stress to communicate the stress signal to the periphery tissue. So next, we would like to first look at where it is expressed. That is easy for us because we can make understanding terms. And we tag the GMP to this protein. And as you can see that is like predicted is mainly expressed on the plasma membrane of neurons, but you can also see some dots within the neurons because the plasma membrane protein is always transported from DR to R into plasma membrane, so you can definitely see a signal within the cells. And also, we'd like to see, we use another marker which could label synapses in the C elegans. Here shows you the red screen. Here shows you that you can see that the TMDM signal is localized really well with a marker labeled synapses. That indicates this transferable protein is not localized on the plasma membrane neurons and also enriched at synapses, indicating it might have something to do with neurotransmission. So next, we would like to see whether it is responding to mitochondrial stress, right? And we a very simple assay that we tested to on up and upon the stress to induce the expression of MM2 from different levels. From the PCR, you can see the NR level is upregulated as well as the protein level. And from the imaging, you can see that both the neuronal level signal as well as the signal and synapses are elevated in response to neuronal modifications. So, it does come to the next question. If it is predicted to have something to do with calcium, and how, right? Because it's not a channel or a pump, so if it's just a small transmembrane protein, how does it function to deal with this calcium? We did this simple experiment because we targeted MMG we do a IP metastatic experiment to identify this potential interacting proteins, and we're hoping to find some lead. And luckily, one of the topics from the metastatic experiment showing that this MCH3 intelligence is a plasmamembrane calcium pump. It's really conserved in mammals. It's actually export calcium from inside to outside. And you can see it scores all the time, so we did a couple of times, and it always comes as a top peak. So we'd like to look into this direction. If MDM2 could all the way calcium with this calcium pump, that really makes sense. So what is MC3 or PMCA? So MC3 encoded gene that is called a plasmamembrane calcium ATPase mediated calcium excretion. That means a actually using this ATP to efflux pump calcium from inside cell to outside. And we know there are a lot of calcium channels and pumps localized on this membrane to maintain the calcium homeostasis in the cell, because neurons not only need calcium to mediate a lot of events, right? But high level of calcium is detrimental. It is to cell death. That's why it's really important to keep a low calcium within the neuron, and that's why this calcium pump is really important. And we were trying to confirm if it's really interactive with this GFP assay, and using GFP, part of GFP targets MDM2 and another part targets MC3. And since that they are always interactive, we haven't done individual assay, but I believe that that's the way that we need to do. And we try to see if we knock down MC3 to probably mimic phenotype of the MDM2. And indeed, when we knock down this MC3 specifically in neurons, they also block this neuronal motility because of mitochondrial communication. And that's since there's a lot of evidence pointing to this calcium regulation, we really want to see what happens within the neurons of these calcium level changes. So we thought this internalized the calcium imaging assay in vivo these evidence, where we using this not quite new, but it's a version that we had at that time, the GCAM6, and we specifically targeted with the plasmamembrane and using the mitochondrial and also the cytosolic, so that we could see calcium changes in different compartmentalized regions. And that means it's really unique because it is transparent, so we could look at a wide individual calcium changes in neurons. And also, we not to mention that we have this marker of synapses, because we really need to look at the calcium changes at synapses to indicate the neurotransmission activity. And here is the data that we got. So we are able to visualize neuronal calcium changes at the subcellular level. Here is just the plasmamembrane at the GCAM, and the below is the GCAM is tackled in the mitochondria. And we confirmed that it indeed works, that we could block the calcium channel. You can see that's the function of mutation of this calcium channel. You can see there's a plus. And also with block the mitochondria MCU level, there is a calcium oscillation within the mitochondria decrease that proved that we are successful making this assay to work. And what's really striking is what we observe that when we have this GCAM on the muscle membrane of neurons in cells, we can see not only there is a visceral level of fluctuation, oscillation happening, that makes sense rather because there's constant neuron transmission activity going on, but in the worms that experiencing mitochondrial disruption within neurons, you can definitely observe there's a flash going on between neurons, even by naked eyes, right? Here you can see that it's flashed several times, and we took a snapshot of this video showing here there's labeling to neurons and constantly oscillate this calcium signal, suggesting, well, something is happening in mitochondrial neuronal mitochondrial function when there's function is disrupted. Calcium membrane calcium actually is changing. And what's interesting is that this calcium oscillation in response to the mitochondrial stress is kind of dependent on the gene that we identify as transferring protein MBM2. So when we block this MBM2, you no longer see this calcium oscillation happening. So that's because of a lot of things that we really look at what happening in the synapses really, because before the video is telling the whole neuron, and we did the quantification of the calcium level at synapses of cystic neurons, and you can see there's we took a snapshot of these 3 points, and so you can see there's oscillation here that this calcium wave is completely suppressed in this MBM2 mutant. And what's really interesting is that when you see that mitochondrial dysfunction induces this calcium oscillation, you can imagine how the cytosolic calcium level is also affected, right? But this is not the case because we did not observe the overall cytosolic calcium level alteration in our case. Here shows me when you keep the mitochondrial calcium is altered because the mitochondrial function is disrupted. That's consistent with our prediction. However, the cytosolic calcium level did not change at all, suggesting that maybe mitochondrial dysfunction could trigger this local calcium oscillation where that occurs near the muscle membrane region of neurons. That's local calcium oscillation happening without altering the entire cytosolic calcium level. So that's completely different from the traditional view of this neuron transmission. And so we'd like to, I think that stage actually makes us to think about the function of mitochondria, not only producing energy and metabolites, but also the function as a calcium buffering machinery, showing here that even though their function cannot be comparable to ER calcium buffer system, but it also could play a role in buffering some of the calcium from the local living neurons that probably decrease the level of this calcium overload. So when we block the mitochondrial calcium channel, we can see this calcium width and also activate mitochondria consistent with our prediction. And so neuron translation, calcium-mediated neuron translation is always coupled with this synaptic vesicle activity. We look at this synaptic vesicles in this MBM2 mutant as well as this mouse mouse neurons that we could find a consistent out there. So definitely accumulation of synaptic vesicles in these 2 mutants. So since neuron translation is involved in this process, that we tried to put serotonin back, and it surprisingly worked really well. It could restore this mitochondrial activation in these MBM2 mutants. And here is something that I related to aging that before I mentioned about this long-term mitochondrial dysfunction, right? Because the ability of mitochondria to respond to stress is the key. And here shows you that this protein actually expression level significantly decreased with age, that indicating the ability of neurons to respond to these calcium changes decreased significantly. Here shows that. And also this synthesis gene is conserved in mouse and animals. the illness as well. The stress level is also changed with age. So we try to restore the neuronal calcium response treatments to see if it could enhance longevity or enhance aging. Delay aging, that's the case that when they overexpress this angiotensin allergens that become already longer. And what's more important, it seems like their neuronal function is doing really well in this ones. So I guess this is our current understanding of neuronal communicative respiratory tissue, and there's a lot to do. And with that, I'm gonna stop here and do a small advertisement for my graduate student that the politician is going to give a poster that on this genetic diversity to modulate the differential effect of aging inhibition. That because the problem that we agree that I think the current aging studies rely on a single genetic model actually limits our understanding of how genetic diversity actually affects aging interventions. That he actually took this project to find this longevity bacteria actually works for a lot of the other strains, but actually really detrimental for some other strains. And that's for one and important for another. So if you are interested, please come to his poster and search through. And with that, I'm going to stop here and thank you for your attention and take questions.